All right, everybody, let's talk about our Q3 exam. So this is the study guide. Hopefully this looks familiar to you. And there's me drawing a little heart. Let me just fix the color here. Hold on. Okay, better. So I'm just going to go through a couple of the sections or the questions you guys have identified as things you want more help with. So I'm going to walk through this with you guys. And then if you have additional questions, always traffic light your study guide. Always color code them. Trust me, it's going to help you. So what we'll do is you can traffic light them and we can bring, you can either email me questions over the weekend or we can spend like a good 5 minutes maybe reviewing. But I think you guys are going to be in great shape. Okay, so let's start with number 9. Here it is, circle. So you're going to transcribe and translate. This, my friends, is transcription. This right here to amino acid is translation. Okay, that's the difference. So what we'll do is we'll go DNA to RNA. So remember that T's are A's and A's bond with U. We replace essentially that there are no T's in RNA. It's all either A's and U's. And I just want to make sure that I am doing this right. Okay, boom, boom. What is real? U, A, U, 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 G, A. Nailed it. Okay. So here's your mRNA sequence, okay? Sweet. Now, here's a little fun fact, and I'm telling you now, and you're probably all going to yell at me. The mRNA, this codon chart, refers to the mRNA sequence. This is your messenger RNA. So this is how you use the mRNA to find the amino acids. Hey, Lighter, how come you're just telling us now? Okay, I think I did tell you. I don't think I did a great job of clarifying. So when it comes to the amino acid part, what we'll do is we'll move up here, giddy up. And so we'll use this sequence and this chart to find the amino acids. And all you do is AUG. AUG. So you go A-U-G, met, right? So it'll start here, met, boom, and then U-G-U, U-G-U. I imagine like a coach yelling that. Is that how coaches yell? Like they just bark at people? Is that how it works? Okay. All right, so U, okay, everyone shut up. I can hear you rolling your eyes at me. G, and then U, right, is valine. And all you have to do then is put, see, I got this here. All you do is have to put the abbreviation for the amino acids. By linking these amino acids, that's when you get a protein, okay? So why do I ask for the tRNA? So the tRNA's job essentially is to run out and get the amino acids. It picks up the amino acids and brings them back to the messenger RNA. So that's why the messenger RNA is the one that codes for the amino acid. The tRNA is just kind of like a go-between. It's like what is it, like an administrative assistant. It's just getting coffee. It's just bringing those amino acids into the messenger RNA, who's the boss of the organization. And so if I ever ask for the tRNA, it's going to be the reverse, right? So again, it's basically going to be the DNA sequence minus T's and U's. So again, I'm going to do the reverse. So if it's AUG here, it's going to be UAC. Right? Does that make sense? Hopefully. So it's basically the same thing, just minus, you just take the T's out and you put the U's. And then that's the sequence for tRNA. So it'd be UAC, ACA, GGCC. God, I hope this makes sense for you guys. If this is making you even more and more confused, hopefully it doesn't. But if it does, please send me an email or come and see me Monday morning so we can kind of chat about this. Hopefully, though, this makes total sense. This is so easy and wonderful. How was I ever confused? Okay, so how many codons? So when we talk about codons, we're talking about these 3 sequences. So you just have to kind of count, or you can count how many bases there are and divide by 3. So you've got AUG, that's 1. UGU, 2. That's 3, 4, 5. So it looks like there's 5 codons. 1, 2, 3, 4, 5. Yeah. So there's 5 codons. Right? Boom. Did I get that right? Whatever. You get the idea. Because you've got 3 bases equal 1 codon. Okay. Cool. Great. Let's keep keeping on. Okay, we're sliding down. Boom, boom, boom. Wait, does this no longer... Oh, my God. Wouldn't it be funny if this doesn't work? Oh, God. All right, I'm going to pause this. Pause. There we go. Back at it. So let's talk about mitosis and meiosis. And I want to give you guys a little chart to kind of compare the two. So the relationship with the number of chromosomes and the somatic cell, I wanted you to know, remember, somatic cells are your body cells. They're not gametes. So they're the same. In other words, when you have mitosis, you have the same number of body cells. The number of chromosomes in a gamete is going to be half that. So you've got, basically, if you've got 40 somatic cells, that's going to mean you have 20 gametes in a gamete cell. And what I mean is 40 chromosomes. So if you've got 40 chromosomes in a somatic cell, you've got 20 chromosomes in a gamete. You have half the number. That's meiosis' job. Meiosis only happens for sperm and egg cells, and its job is to give you half the number of chromosomes, okay? So when we talk about our chromosome, I want you guys, hopefully, here we go. We've got like our little butterfly. So that's our chromosome, okay? In the center, you have your centromere. Okay. Great. You have your chromatid. Which in your chromatid is like, here's one chromatid, and this is the other chromatid. You call them sister chromatids. That's basically it. It's basically all you need to know. Remember that it's made up of chromatin. Chromatin is just DNA in a protein. And we call that protein a histone. Sweet. Cell cycle, the phases are going to be, just so we know, interphase, mitosis, and then let's say the S phase, and the S phase stands for synthesis. So this is going to be interphase, I for interphase, M for mitosis, and S is synthesis, and that's when the DNA is replicated. Great. So let's just take a quick momentarino, as you might say, and let's just do a mitosis versus meiosis chart. God, if only there was some like really cool science teachers that did a mitosis and meiosis rap battle. That'd come in handy now, right? Okay, part of the reason why I show some of you guys those videos of teachers rapping is so you appreciate how cool I really am. Okay, mitosis, right? You're going to have a clone. So it's going to be genetically identical. Right? Meiosis, nope, genetically different. Because of something like crossing over. Okay, you've got your chromosomes crossing over in prophase 1, and you have this exchange of DNA. So you're going to have a different sequence. They're going to be genetically different. Mitosis, you're going to get 2 cells. Meiosis, you're going to get 4 cells. You're going to have your sperm and your egg are going to be meiosis. These mitosis are going to be your body cells. The second main difference is you've got your PMAT. You've got your PMAT, your phases. They split once. Meiosis, it splits twice. So you've got your PMAT, and then you have your PMAT2. okay? And that's because your goal here is 4 cells, not 2. You want 4. Sweet. All right, let's just talk about genetics really quickly. So trait and allele. Trait is going to be your phenotype. That's your physical. What something looks like. I spelled physical wrong. Don't judge me. The allele is going to be part of your genes. It's your genotype. Okay? That's all that is. An example of a phenotype would be like green eyes, which is what I have. A genotype might be this. I don't know if that's the actual genotype, but just go with it, all right? So that's the only difference there. Let's look at this. Let's just look at this one really quickly. So if you have black hair is dominant to red hair, okay? So use the following genotypes and phenotypes. Homozygous dominant is going to be this. Boom, boom. Homozygous recessive is going to be here. And this is going to be heterozygous. The phenotype, you're going to have black hair, because that's dominant, right? And this is going to be red. Hopefully that makes sense. Let's talk about these crosses. So now, because I'm the next human being alive, there is no dihybrid cross that you have to complete necessarily. There's one where I think I give you some information, I have you ask a question, but you don't have to actually do a dihybrid cross. So let's just kind of skip around and let's just spend some time on the blood type, because I think if you can do 7, you can do 6. So you've got these blood types, right? And they're codominant, which means that AB, they're both dominant alleles and they're both expressed, okay? So let's figure out what we're going on here. So for AB, we're going to use I, because that's the letter we use for blood type. This is B and this is A, okay? For O, it's recessive, and remember, in order for a recessive trait to be shown, the genotype has to be homozygous. So here we go. So we just go boom, 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 boom. Blood type. Have I mentioned to you guys that I am an A+? My blood type is an A. My blood type is an A+, just like me. A+. Okay. So there we go. Hopefully this makes sense to you. Also, when we talk about pluses and negatives, that's actually something called the RH factor that we'll talk a little about when we talk more about anatomy and stuff like that if we get to it, which I think we will. Anyway, so here we go. So what are the phenotypes? You're going to have 50% are going to have blood type A, and 50 will have B. Okay? Cool. That's basically it. Pedigrees. So what are we looking for pedigrees? I'm basically looking for you to be able to analyze them. So know that men are squares, circles are women, okay? The way I believe I have it on the exam, and I'll double check this, is that this is a carrier. All right? This means you're a carrier. Now, it can also mean this if it's not a sex-linked trait. Men can also carry if it's not a sex-linked trait. This means you're expressing it. Okay? That means the trait's expressed. And this is what we call the norms. Normal. I hate that we call it that, because what's normal, everybody? So that's what that's all about. Marriages are denoted by this. Children are like this. Okay? That's basically it. When we talk about Mendelian, we're talking about this very simple dominant and recessive. One is dominant, one is recessive. The dominant takes precedence, end of story. So it's this idea of complete dominance. That's a Mendelian genetic, right? So the non-Mendelian are things like incomplete dominance or codominance, where it's in between. These are not on the test. Boop, boop, beep, beep, bop, boop. And epigenetics, guys, remember, epigenetics is that idea that there's a protein or methyl group, I should say, on the DNA strand, and that controls which genes are turned on and off. It does not in no way, I almost said it does not in no way. It don't do that. It does not change the genetic code, the sequence. It just changes the expression, Okay, so epi above genetics, the genome, the DNA. All right, party people, that's what I got. So hopefully this helps to clarify some things. Please email me questions, bring in questions. You can stop by, see me tomorrow or Friday after school or Monday morning. Lighter out.